we'll have the final 10 minutes for program consultation. So it's going to be an open floor discussion, question and answer. And by the way, through Zoom, we have a chat option. So if you actually look at the bottom of the screen, there is a chat option. So by all means, type in your questions to the chat channel, and then we'll try to answer those questions live. And then I'll pick up some questions towards the end and give everybody an answer. So without further ado, let me we'll quickly move on to the taster sharing. So I'm going to share the screen, what we call a course document. Now, this is actually a document that each course in university will, in our university, will have. So it basically describes the content in terms of keywords. So you can see that the yellow part has been highlighted, which basically are the keywords of the contents. So since this is actually biomedical engineering lab, and it basically provides training on our biomedical engineer graduates on how to conduct laboratory work. Now it has two parts. One is called the wet lab, which is concerned with biology, microbiology, DNA amplification, and so on, and also lab safety, okay? And then the interpretation of data, technical writing, i.e. report writing as well. And then the second part will be on the building hardware, particularly electronics, and then how the electronics can be used to solve biomedical engineering problems, okay? And then well, our university is bilingual, so each course document has both the Chinese and the English version, but predominantly the language that we use in our faculty, engineering faculty, we use 100% English. So, and then you can see also the blue highlighted, what we call learning outcomes. It's a key component in the course document in which students are expected to acquire these skills after they have finished the course, okay? So I'm not going to go through the details, but you can see that, you know, all the contents have special objectives of training students for a particular skill set. So I'm going to go through to the next part. Now, in this course document, it actually highlights the 13 weeks of activities. In this university system, we have 2 semesters every year, and each semester has 13 weeks. Now, this one was actually for last year's teaching. So you can actually see the dates, and you can actually see all the way from week 1 to week 13, all the different activities. And you also see the venue. Now, university education is different from that of a school. It's very sort of a lot of autonomy. Students are expected to show up at a classroom. So each course has a different classroom. So there is a venue, and then there's also a timetable, and you know which day, which hour of the day that this course will take place. And then each week has its course activities, and you can see there are 2 parts. The first part is what we call the wet lab activities. Okay, wet lab activities. In other words, you know, we train students how to go to lab and actually conduct lab. And that's actually been looked after by Professor Li Ting-Duan, who is right next to me. So after I finish this course document, I'll hand over to Professor Duan. And then the second part of the lab, which is actually highlighted in green, which is what we call dry lab. It's concerned with microcontrollers, computer-controlled hardware. And in this year, we are asking students to build a heater device, a constant heating device to do DNA amplification for detection of a specific DNA. Now, let me take an example. For example, the COVID-19 diagnosis. Okay, so the nurse takes a deep throat sample or saliva sample from the patient, and that sample will go through what we call DNA amplification process to detect certain fragments. And this is what we are trying to do. So we're asking students to actually build an electronic heater platform to perform that job. So then this is actually looked after by Professor Wu for that year, and this year is looked after by me. So we have lectures to train students about the electronic components and the microcontrollers, Arduino, which we can program to perform the job, and also some of the mathematics behind temperature control, a control system, let's put it that way. And then they need to write a report. So then let me continue. And then, the assessment schemes, with different marks goes to different parts, different components of the course and so on. Okay, so I'm going to quickly switch over to Professor Duan, since she's actually looking after the wet lab parts. So I'm going to actually hand over to Professor Duan for the wet lab course components.
Okay. Thank you, Professor Ho, for the nice introduction. So actually, for the major of biomedical engineering, is quite experimental in nature. That means you need a lot of training in doing experiments, either like testing or making your own devices. So for this purpose, so in the whole undergrad curriculum, so we have courses specifically designed for those experimental training, or some courses integrated with both experiments and the lectures together. And the course Professor Ho just introduced is the BME 2011. That course is a 100% experimental training course. So, and as you can see that just from the name, biomedical engineering, so bio is one of the important part. So the part I was in charge of is more focused on bio. And the bio, you may know that it will have different biosystem. So microbe is one, mammalian is one, and as an introductory training course, so we mostly focus on microbe. And our department has a relatively large teaching lab with all the essential bioequipments accommodated in it. So next I'm going to show you the teaching lab. We made a lab tour video, and with all the equipments described and explained. So let's just take a look at the lab tour video.
Oh,
audio?
So sound.
Audio. Yeah. Will provide a video tour to our department's teaching lab. So this is our department's tour.
Louder.
Moving with engineering building. And the teaching lab is to our right, to my right hand. So this way. So this is one of our entrance to the teaching lab. 1102 is the room number for our teaching lab. So let's enter this teaching lab. And upon the entrance, we have another room just at the right-hand side, which is our BME Technicians Room. So you can find our technicians in this room, Dave, Kane, and Nelson. And let's come this way. And this is our teaching lab. So you can see a lot of stuff here, electronics and equipment. So basically, we have two lines of benches. One is L-shaped. The other one is just a straight shape like this. And then in the next several minutes, I'm going to introduce some of the important equipment to you that you might use in the future. So come this way. Our microwave. So, but this microwave is definitely for experiments. No food, no drink. We usually use microwave to heat up water solutions. So this is our microwave. And this is the ultrasonic cleaner. So it will help us to use ultrasonic waves to clean the surfaces of glasses. And we have this autoclave machine. So it will generate high temperature and high pressure to sterilize everything inside this equipment. And it will be used in our first several labs. And here is the water system. So it will filter the water, so it will become ultra pure water. And then this water can be used for different kinds of cell cultures. And we have this. It's called chemical hood. So you can see this chemical hood is very special. It has a very big ventilation system connected to the roof of this building. So everything, the air will be circulated inside this hood, and then eventually will be exhausted to outside this building. And because of the good ventilation, if we have anything that's volatile and that's smelly or somehow dangerous, we need to handle it inside this chemical hood. And this is our sink. We use it to wash different bottles here and then dry it in the racks. And we also have the ice machine. So it will have a lot of flake ice in it. So when we do some biological experiments, we need low temperature, around 0 Celsius degree. We may get some ice and put in a container to sustain a low temperature in a outside environment. And this is our BSC, so biosafety cabinet. So when we handle some cell cultures that require very clean environment, we will need to turn on this equipment and handle everything, every samples inside this equipment. And of course, everything before put inside this equipment, we need to sterilize it by different ways. For example, autoclave or by ethanol spreading. And maybe you can just give an overview of this lab. And then I'll leave it. Okay, so that's basically our teaching lab. So we are looking forward to you joining the laboratories in a few days. So we also have a few pieces of equipment.
So just now we watched the introduction of our teaching lab, and then next video we are going to demonstrate one of the experiments that use several reagent kits to extract DNA out from the microbes sample. So particularly because of the COVID-19 situations, you may know that analysis of genetic information is really, really important for us. So you can, from the next video, you will see one important way, one popular way how we extract DNA from the bacteria.
Now we're entering part B of our experiment. In this part, we'll be measuring the DNA concentration of the DNA that you just extracted and purified. First, let's make sure that you've prepared all the materials necessary. You will have to prepare a sterile elution buffer, in this case, water, which is the same elution buffer that you used to extract the DNA in the previous part. And then prepare the P2 pipetta. The sterile pipette tips, clean wipe, and the DNA. Also, make sure that you open the nanodrop. The nanodrop will be the main machine that we use to measure DNA concentration. As you can see, it has a control panel on the top, the lever arm, and a place where you put your DNA sample. Because we're measuring double-stranded DNA, we first choose dsDNA on the control panel. and it'll instruct us to load blank, which is the water that you prepared. First, pick a piece of Kim wipe, lift up the lever arm, and clean both pedestals, I guess, to ensure that it's perfectly clean before you load any solution on. Then, turn the pipette to 1 microliter. Take 1 tip and take 1 microliter of blanking solution, which in this case water. Be very careful because it's a very small amount. So look at the tip to make sure that you actually load on the tip some of the solution. Load the blanking solution on the pedestal very carefully. You can do it very slowly and you can use your left hand as a support. Do not let the tip touch the pedestal. And very carefully load the solution on. And then load the lever on, and it will start blanking. After the blanking is completed, lift up the lever arm and clean both the lower and upper pedestals with a Kim wipe. Just touch it with the Kim wipe to make sure that all the solutions are absorbed by the tissue paper. Then we're going to load your DNA sample on the nanodrop. Still 1 microliter. Pick up a new tip. Take your own DNA sample that you extracted and put out 1 microliter. Just like how you put on the blanking solution. Load your DNA sample very carefully on the pedestal. Load on, and it will automatically start measuring. Here you can see the result. It will show you a spectrum of light absorption of your sample. And on the top right corner, you will see the measured concentration of your DNA and some other. Check your spectrum and see if it is the typical absorbance spectrum of DNA sample. Also, look at A260 to A280 and see if the number is correct or approximately right for a pure DNA sample. Sometimes it will be a little bit higher than 1.8. 1.8 to 2 is the acceptable range. Then, on your DNA sample, you can use marker pen and record the concentration. In this case, it's 101.8 nanograms per microliter. Sorry about that. And again, each time you finish up with measurement, lift up the arm, take a Kim wipe, and make sure to clean all the pedestals after use. And then you can press and experiment if all the experiments are done. you already cleaned the sample area, you can lower the arm and touch continue, and we'll be back to the main page.
Okay, right. So now that you've seen the videos of the first part of the lab, and the aim is actually to extract DNA from bacteria. Now, the next question we want to ask is, what identity are those bacteria? What kind of bacteria are they? So we do some kind of DNA fingerprint test. So the second part of this course teaches how can we actually do DNA fingerprinting. We use a technique called DNA amplification, very akin to the COVID-19 test that people nowadays do. Now, I'm going to share the screen for the second part of the laboratory work. Now, the key behind DNA amplification is a process called polymerase chain reaction. So this is actually part of the nooks that I use for my part of the course in the second part. So the polymerase chain reaction seeks to use a reagent in which the sample goes through temperature cycling between 55 degrees to 93 degrees. And then as the goes through temperature cycling, then the DNA of the target species will be amplified. In other words, the population get increased, and then we can actually detect them because you're talking about a very small amount. Now, an alternative way of doing it is called loop-mediated isothermal amplification. It can do the same job, but the temperature required is constant. You don't need to change the temperature, and in our case, it's 67 degrees C. So the next question is, how can we do that? So then, in our laboratory, we can actually So in the second part of the course, 7 weeks, we're going to give our students, each one of them, a set of kits, okay? So we give them these components, a small microcontroller board and a heater block, and then some resistive heater devices, and then some keypads for inputting numbers, and then we ask them to actually generate a constant temperature in this heater block. And that heater block will actually heat up the sample. As you also saw the Attendorf that Professor Don used for keeping the DNA. So if the target DNAs are there, then it changes color. And then we know that, this is the bacteria that we're looking for. So the next question is, how do we actually build that hardware, which includes programming the microcomputer from a computer, heating, and then detecting the temperature, feed the signal back to the board, and then analyze the temperature to see if it actually reaches the target number, and then keep it constant, so that by looping the signal continuously. So that's basically what it is. And then towards the end of that part, we'll actually set out some conditions for students to test their prototype, like blowing wind to the fan, blowing wind to the device to see if it still maintains the temperature. If it drops, then we deduct some mark, and whether or not it will actually maintain temperature within the specific time, and so on. So a lot of it actually is to implement programming skills and implement mathematics behind it, and build the software, and then program the microcontroller to the job. Now, university learning, is very much self-managed. So we only give students the tools, we only teach them the skills, but we don't actually tell them the process. So this is actually the end of the 7 weeks, that each student come back with their own prototype and then go through a laboratory test to see if their device are doing the job or not. Now, every week, we actually give students a lecture on giving them the skills. So I'm going to show you some sample lectures. So now, for example, this is actually note number 2. Okay, so in 1 week, I would actually tell students aluminum block, and then the power control, temperature sensors, and then the microcontroller hardware called Arduino, and then different components of the system. And then also the mathematics behind temperature control. So I'll tell them what are semiconductor devices, what we call pulse width modulation for controlling power, and then the devices involved, and then the examples of using such techniques, and then some of the circuits involved, and the devices involved, and also the temperature sensor called thermocouple, and so on. So students learn the physics, the actual devices and their characteristics. And then with those knowledge, they go off and find their own plan and work on their own pace to deliver the prototype. And then in another week, I'll tell them about the mathematics behind real-time feedback control, how data are collected by the sensor and then being processed by the computer with what we call the PID, proportional, integral, and derivative approach, and then generate the output for the heater to maintain the temperature. Okay, so and then similar techniques are being used in shock absorbers in automobile industry. And then the actual circuits. So students actually need to build these circuits and then tune the performance of those circuits. Now, at this point in time, you may not be aware of the components inside. They look very sort of alien to you, but it doesn't matter because, when you are at that level, when you're in the lecture, then you'll be able to actually see what they are for. And then the everything behind it, and also some of the very complicated math behind them. And don't be too scared by it, because again, when you're at that level, 2 years later, when you have been properly trained, you'll be able to actually appreciate these differential equations. Because actually, in year 1 and year 2, we have 2 mathematical courses by the engineering faculty to actually tell students these differential equations and their applications in, for example, resonance circuits, in shock absorbing mechanics, and so on. Okay, and then their characteristics. So these are the example of the contents that we teach in this course. Now, without further ado, I think we are more or less overshooting time. So I'll wait until you actually raise further questions from through the chat room, and then see if we can actually answer your questions. So I have presented you a squeezed version of 13 weeks worth of content of a course from BME, and I hope you sort of find some use of it. And without further ado, I'm going to hand over to our student ambassadors to share the experience with you. Now, without any pre-assigned order, I'm going to ask Cassie Jung to start off sharing her experience of learning in BME. So let me hand over to Cassie.
Thank you, Professor Ho. And good morning, everyone. I'm Cassie from the Biomedical Engineering Department, and I'm currently studying in year 2. And me and Vicky are also studying this course right now. And so actually we are going through what the professors just told us, the contents. And this course, I think, is a very fun course because it's a very unique experience for us to have both experience from doing the bio part of the lab and the electronic engineering part of the lab, which is referring to the wet lab and dry lab, which normally from other departments or other majors, you usually only get one part of the training. But because as we are from the biomedical engineering, we require both knowledges from both fields. So we are getting experiences of actually getting to do two very different fields of experiments. And for us, it's a little bit unfortunate because due to the pandemic, we lost some hands-on experience on doing the lab. But thanks to our professors and technicians, we are still able to get to learn things from videos. And for the dry lab part, the department actually gave us the lab tools for each group to continue to proceed their projects, which I'm actually now doing it. And it's quite challenging, I would say, but because it requires many knowledge and skills from different fields, just like Professor Ho mentions, we'll need knowledge from the biology part and programming and physics. And I think it's very interesting of doing this kind of integrated tasks. So maybe I can hand over to Vicky to do some other sharing.
Okay, thank you, Cassie, for the nice introduction. Okay, to all of you who are thinking of BME for your undergrad study, so I'm here for you with some experience to see if it's attractive or persuasive enough for you. So when I was having my high school, when I was studying, when I swallowed the whole biology textbook and physics, and I always thinking of what can I do with it, and because the knowledge seems so scattered and I want to combine them to do something practical. So I think BME is the right place for you to go. For example, I have some fun courses, which is me and my group doing a project to using a whole semester, using 4 months to create products. We're doing a blood pressure sensor, something like that, and it feels really satisfactory to incorporate your knowledge with to do something really usable. And the other fun thing of BME you have is that there is a lot of non-local students for you to broaden your horizon. For example, in 2 years of study in BME, I met people from France, Kyrgyzstan, Bangladesh, Korean, Japan, and they exchanged their cultures with me, not only in the academic level. We share a lot of experience, we learn from each other, and it was a really fun experience. And recently I participated in international buddy scheme, which is all about me and my partner from a non-local, which is a non-local student. We work together to solve a problem internationally. We are working on a dengue fever, how to solve the dengue fever problem in Bangladesh. So we are making a poster, we exchange ideas, we're having dinner. So it was a really great time for you to have in BME. So consider it. So if you have any questions, you can ask in this following minutes.
Hi.
Thank you, Vicky, and thank you, Cassie. Now, we are going to actually move on to the next part of the event, which is actually open floor discussion, Q&A. So I understand that amongst you, you may have questions that you would like to ask, and you may actually want to get to know more about the university and BME, the program, and then other aspects of university education. So please, either you actually ask the live questions on the phone, or you actually can type in your message. So now the floor is for everybody.
Hi. What IT knowledge do we need to have before studying BME?
IT knowledge.
I didn't study IT in our secondary. I didn't study IT as my elective in our secondary school. What IT knowledge do I need?
I think everybody knows how to use computer, and as Professor Ho just mentioned, actually there's a lot of projects involving using programming. So It is not required, but it will be better if you actually must all have a little bit knowledge on certain computer language. For example, MATLAB. MATLAB is very useful for solving mathematical problems, and also C++ for control some instruments, and also Python. Particularly Python is very popular right now due to the artificial intelligence getting increasingly used in a biomedical imaging field to basically processing the biomedical image, for example, MRI and the CT image. So I think those languages is pretty important for our major, but it not require you master all the language. Probably one or two will be actually good enough.
No, as of now, I don't know anything right now.
Right. So if I may actually sort of also chip in some of my comments. Now, one thing I must stress is that biomedical engineering, by its nature, it's very multidisciplinary, very diverse. It involves a lot of skills. Now, that actually is a pro as well as a con. The pro is that it is addressing a very current issue. I mean, healthcare is a very current issue. And in the foreseeable future, the world needs a lot of new technology to address those issues. But the con is that by its nature, it's very multidisciplinary, meaning that it involves a lot of expertise. Now, in your case, you have not been exposed to any IT training or a lot of IT training. Now, this situation does occur amongst the students that we have admitted. Now, the university, the engineering faculty, actually provides IT training in their year one course selection. Now, likewise, for example, if somebody who actually didn't have biology, because in fact there is another question being raised in the chat room, that is biology a important subject? It's actually the same situation that as compared to IT. So another student who actually get admitted may not have a lot of biology exposure before coming to our program. Then again, in their first year, students have the option to actually pick a course that trains a specific skills, namely biology. So that student I very much encourage he or her to take a biology course in their year one. And then the same, likewise, you should actually take an IT course in the year one, so that you can more or less get yourself, well equipped to deal with the program contents. And we actually admit students who have quite diverse backgrounds in light of the situation that we are encountering. It's not like, for example, computer science. Everything is computer. Everything is programming. It's not like mechanical engineering. Everything is physics, maths, and material science. BME, it's extremely diverse, right? Yeah. Professor Yuan came back from Johns Hopkins. He's been working in optics, photonics, fiber optics for detecting tumors in the brain. I come from EE background. I've been working on using light, photons, and lasers to detect molecules. And Professor Duan, you come from
chemistry. I come from chemistry background, so I got my undergrad. I got my bachelor's degree and a PhD degree both in chemistry. But now I'm working on a more biology topics, biology field.
Biochemistry.
Yes.
Yeah. Mm. So I hope I've answered the question. So thank you very much. Right. Any other questions? Any other questions? not the client this time.
Yeah.
Maybe I can supplement a few sentences on the question sent in the chat. So I know some of you may not take biology courses in your high school, but as Professor Ho just mentioned, our students as well as our professors are coming from different backgrounds. So for those who don't have much knowledge in biology, or for those who don't have much knowledge in physics or other subjects, so our department actually provide the enhancement course, right?
Mm-hmm.
We'll continue to do that. So we are doing it for the last several years. We are going to do that. So the enhancement course will be provided on weekends, and then we are inviting students, undergrad students or postgraduate students, to come together with professors to provide additional teaching, additional sharing on specific subject, like biology for one term and physics for one term. So in addition to normal learning, you will have extra resources to enhance some subjects you feel you need more learning.
Yeah. Okay, so also to supplement a little bit more, the idea of having BME student ambassadors is also something along that line too. We actually try to create a scheme that senior students may be made available with resources from the department to help junior students in terms of guiding their academic progress as well as settling in the university and so on. So okay, so we have more questions. So will we
have a placement at
hospital so
that can be available? Yeah.
Yes.
We have one course called biomedical engineering
and hospital experience.
Yeah. And we also providing internship.
Yeah.
During summer.
Yeah.
So students will actually work in the hospital.
Yeah. Yeah. So there are two types of placements. One is called work study, and it's a gap year. Students actually go out for 12 months to work in a company who is actually in the BME industry, and then it's a full-time salary acquiring job. So it's a gap year. Usually take place after students finish their third year before coming back for the fourth year. The other type is summer placements, and both the government EMSD, the electrical and mechanical engineering department, actually hire internship from the department. So we send a few students to government for summer training. And also some of the hospitals in Hong Kong, private and health authority ones, give us summer training as well. And then what else?
Let's take another question. May I ask, if we study BME, what jobs could we consider? Okay, so this question: if we study BME, what jobs could we consider? Well, the graduate employment destination, as far as I can remember, has three categories. One, joining the biomedical profession as biomedical engineers in hospital, both public and private. Biomedical engineering for selling commercial products in the healthcare industry. So they interact with hospitals, but from the supplier side. And then another group goes to further the studies, either PhD or MPhil. And then another group goes to work in the government, in different departments. And another group, they go to others. That is, they can, they do all kinds of jobs using their degree training. And amongst them, the biggest proportions are those who actually work for the biomedical industry, both from the user side as well as the supplier side.
Yeah, so that's the statistics of our graduates' employment destination. So if I have more...
If I have more interest in working in lab, am I still go studying BME? Lab? Hospital lab? Yes. I mean, either the research lab in the university, company, or hospitals. So as long as they are biomedical related, I think BME is a good place for you to study.
So maybe I can contribute more information about lab experience in BME. So actually, I would say definitely yes. So BME is the maybe one of the best choices you can find on campus if you want to have more laboratory experiences. Because as you can scroll down our BME's professor webpage, one thing you can find is that we have a lot of young professors, and we have a very dynamic and energetic environment in our department. So we really encourage all the undergrad students to actively seek for research experiences from the first day they start the undergrad program. So you can just knock the door of each professor and ask, hey, whether I can join your lab and do some laboratory experiences. So this is what I have encountered many times during the last few years, since I have already joined CUHK for 3 years. But many undergrads just ask me for this kind of opportunities. And in addition, so from the first day, you can just ask whether you can join lab. We also offer undergrad research courses, so you can officially enroll in this course, earn some credits, and then officially do the research in professors' lab. And that's one official opportunity. Another thing is we offer undergrad summer intern. So it will be a 10 weeks duration and funded by the faculty. And then you can do research in whatever lab you want, but it's application basis. And at the end of the summer intern, you will have some chances to present your work. So another official opportunity is the final year project. So this will last around 1 year. So in the 4th year of the study, you can choose your supervisor and then do a 1 year project in that under that professor's advice. So we do provide a lot of opportunities for you to really merge inside the research and laboratory environment. Yeah.
Professor Yuan, you have anything to
I just found there's another question.
Yeah, so let me finish this question. While Professor Duan was speaking just now, I was sort of going through on my mind how does BME in terms of laboratory activities compared to other programs in university. Then immediately came to my mind was chemistry. Now, in fact, if anybody would probably think that chemistry has a lot of labs to do. Indeed, they have. I think BME doesn't have as much laboratory work as chemistry, if I may say that. Yeah, because but chemistry is very specific. You have a lot of glassware, you have a lot of chemicals, and you're dealing with chemicals and glassware all the time, Bunsen burner all the time. Whereas in the case of BME, which I guess will probably have less laboratory comparatively, the kind of labs that we deal with, as much as I can think of, are DNA extraction, DNA amplification, DNA detection, virus detection, and then in my case, the building prototypes using 3D printers, building the devices for light detection,
CTC detection.
Yeah, and then circulating tumor cell detection. Yeah. And then another colleague, he might be actually involved in medical robotics. So meaning that they'll be building mechanical robots, also writing software to drive those robots for performing the functions that are medical related. In that case, it's rehabilitation. Okay, so sort of movement assisted robots. So I think those are the labs that we're involved. So I think that's as much as I want to say. So let's move on to the next question.
Learning about neuroengineering.
Should I study BME if I want to learn about neuroengineering? What is the reason?
So that's a very good question because we do have a course. The course name is exactly neuroengineering. Yeah, and in this course is a mixed course mixed by lectures and also experiments. So you will learn neuroengineering from two sides. So one side is from the electronics ways, so how to use the devices to stimulate and to promote the neural activities for neuromodulation and then for neural treatment. So the other side, other perspective is from the biology side, from the biological mechanism. How do we understand the mechanisms underlying the neuron regeneration, neural injury, and neural repair? Yeah. So you can see that neuroengineering indeed is one important topic we have in BME, and then combining all the expertise from professors in BME while trying to provide different angles so you can comprehensively to understand this topic.
So I look at my watch. Since time is actually running out, I would like to actually invite the last question before we end the session. Of course, I mean, while we'll stay on for a little while, but at least we close this session, and then more interested students might still want to stay on and ask this question. But before we formally close this session, I would like to invite the last question. No? Let's see. Any other questions? Any other questions? All right, then, so I assume that, there's no further question. Then I'll formally close this session